Hello guys, today we are going to discuss about a blood test called peripheral blood smear or blood frame. Use of the test to find out if red blood cells or white blood cells and platelets are normal in appearance and number. To distinguish between the different types of white blood cells and to determine the relative percentages in the blood. To help diagnose a range of deficiencies, diseases, and disorders involving blood cell production, function, and destruction, to monitor cell production and cell maturity in diseases such as anemia, leukemia, during chemo or radiation therapy, or in evaluation for hemoglobin variants. A blood film allows the evaluation of a white blood cells, red blood cells, and platelets. These cells are produced and matured in bone marrow and are released into the bloodstream when needed. WBC main function is to fight infection, while RBC carry oxygen to the whole of the body. Platelets appear as a small cell fragments and when activated stick together to form a plug as one of the first step to stop bleeding. The number and type of each cell present in the blood changes but is normally maintained by the body within specific ranges. Values can change at times of illness or stress. Intense exercise or smoking can also affect cell count. A blood film is a snapshot of the cells that are present in the blood at that time that sample is obtained. To produce a blood film, a single drop of blood is spread in a thin layer across a glass slide, dried, and then stained with a special dye. Once the stain has dried, the slide is looked under a microscope by a medical scientist, medical laboratory technologist, or hematopathologist or hematologist. A drop of blood on the slide contains millions of RBC, thousands of WBC, and hundreds of thousands of platelets. Under the microscope, the stained WBC can be easily seen and counted to estimate the number of each type of a cell present. In addition, the size, shape, and general appearance of the cell can be compared to that of a normal cell. It is possible to distinguish between the five different types of WBC: neutrophil, monocyte, lymphocyte, basophil, etc., and to find the relative percentage by counting 100 consecutive cells. During this examination, the size, shape, and color indicator of a hemoglobin count of the RBC can be measured and number of platelets estimated. Sample required is blood. Normal range depend upon lab to lab, that is about PBS.